when Liz and I started to collaborate, the effort there was to see if what they had learned and would learn in tetrahymena would be more broadly applicable, would apply to other organisms like yeast and later on people.
So Jack sent the two types of DNA to me. These were the linear DNAs that had been living in yeast for many generations. And I was very excited to understand what might have happened, if anything, to the tetrahymena telomeres that had been living in this foreign environment for many, many cell generations. And also, what was the nature of the piece of yeast chromosomal DNA, which looked like it had come from a chromosomal end, from a yeast chromosomal end. What was the nature of that DNA? And I was very excited to find out what would its DNA sequence and structure look like. First of all, as one read the sequence, reading from the internal part of the DNA out toward the end, first of all, very reassuringly, we could see our old tetrahymena sequence. 2T building blocks, 4G building blocks, 2T, 4G, on and on, right, for tens of these repeats. And then suddenly, as one read the DNA sequence, it suddenly changed. And suddenly it was now very different. It was actually made up of 1 or 2 or 3 Gs, and then there'd be a T, and then 1 or 2 or 3 Gs and a T, and this was really different. But the immediate thing that jumped out was that it was all made up of just the same 2 building blocks. So that the sequence was alike, and yet it was different. So that was very exciting. And then the sequence that came from the end of the yeast chromosome was made up of this 1 or 2 or 3 Gs in a row and separated by individual Ts. And it was just irregular. It didn't have any strict repeating pattern, but it was always made up of these motifs. The yeast telomeric DNA sequence was joined directly onto the tetrahymena telomeric DNA sequences. So here was now some really interesting threads coming together, because there was that result, and there was no way that the yeast sequence could have obeyed what we thought were the normal rules for DNA molecules joining together and stuck itself onto the tetrahymena sequence. That just wasn't in, wasn't in the manual for DNA. What we were doing was saying, is there an enzyme which acts on DNA which has never been seen before? So that was the question that this work left us with in about 1983 or so, when we had the sequence information of the tetrahymena telomeres after they'd been living in yeast, and of the yeast telomere.
So I was involved in that story, in that work for several years, our initial collaboration. And then we diverged as Liz's lab went in the more biochemical direction, and with her student Carol Greider, they actually found the enzyme that had been predicted from our earlier collaboration.